I found him. Hey, Laurits.
Nice to meet you.
Nice to meet you.
In person this time.
I know. Sorry, I'm already filming. Laurits Skov, geneticist and legend. Third time on the channel.
Thank you for having me back. Thank you for having me back. And welcome to Berkeley.
Obviously, from that clip, you can tell that's Laurits Skov, and we're in Berkeley. He is an expert at Neanderthal genetics, ancient DNA. He's already appeared on the channel twice. He's an absolute legend. And I had the opportunity to go down and visit. And of course, I had to say yes. We had a really interesting chat all about how ancient DNA works. I'm always so curious when big scientific discoveries come out. You know, what are people looking at on the screen when they're talking about these things? Do you know what I mean? Do you ever feel like that? Like, what are they literally seeing on their screen when they say, oh, wow, this is related to this, or what have you? So we had a great chat about that. We also discussed how he's hoping to find new archaic species. Well, not new, I suppose. They're long dead, but new to science that we have potentially interbred with. And yeah, honestly, I learned a lot about ancient DNA. I really, really learned a lot. It's very cool. I love it so much. I'm obviously extremely fortunate to get to go and pursue my curiosity and ask interesting questions of scientists. I'm very, very lucky that I can do that. And it's because of you guys watching, and it's because of KiwiCo, the masters of curiosity, paying for this thing. So I just want to thank them now. KiwiCo's mission is to get your kids learning about the world around them. And honestly, I'm extremely proud to be sponsored by them. I love that goal. Love that mission. My kids love being in these KiwiCo clubs. It's the new name for their crates that my kids have been getting for over a year. They absolutely love them. They recently got this fun space exploration one. And another one that was experimenting with colors. Super well made, very high quality stuff. They're for different age groups. My kids are young. These ones are for different age groups, but honestly, they both play with them together, which I love, and is not always easy to do as a parent, as you probably know. They really dive right into these activities. You know, as soon as the box arrives, it's go time. I can definitely tell that the subject really sinks into their brains, because later on, they'll come up again when we're doing a different game. Like in the evening after this colors one, now we're using the things as magnifying glasses, and we're color matching detectives looking for the carrot thief. I don't know where the carrots came from. That wasn't in the KiwiCo box, but still, you can see that the learning is sinking in. My kids are young, but if your kids are a little bit older, KiwiCo is super excited about their KiwiCo labs. They're really designed to not just be one thing that you just do one time and get rid of. They're designed to be for continuous use and build on each other so your kid gets a good understanding of science. If KiwiCo are excited about them, then I'm sure they're going to be great, because my kids love all the KiwiCo stuff. But yeah, I can't emphasize enough how much I want my kids to be curious about the world around them, and KiwiCo is great for that. I love it, and I'm very proud to be sponsored by them. If you feel that that's also important to you, go to kiwico.com/Stefan to get 50% off your first month. And yeah, let's get chatting about ancient DNA. Which way are we going?
We're going up here.
This
is where I work at Standing Hall.
Oh, nice.
Have you seen this?
But reserved for Nobel laureates.
Yeah. You gotta have a Nobel Prize if you wanna park here.
And then this is where we clamp just to sync them all up.
Oh, yeah, like they do in the movies.
That's what they do with the sound so that all the microphones, you can line them all up at the correct point.
Can I do the clap?
You can do the clap, go for it. There we go. Perfect. Yeah, so I thought maybe a super basic question to start.
Yeah.
What is DNA, and what are these 4 letters that we're going to be talking about?
Oh, yeah.
Start from the very, very beginning.
Yes. So in your cells, there is DNA, this double-stranded helix thing, and it is a molecule, and it's like really long. It's maybe they're arranged into chromosomes, but it's just a long strand of different types of base pairs.
Yeah.
That are pairing to each other. There's just these 4 different types of chemical compounds. We call them bases.
Yeah.
And they are called adenine, thymine, cytosine, guanine. So we could just abbreviate that to A, T, C, and G. And your DNA is just you can think of it as a long book just written of those 4 letters repeated.
Yeah.
And there's like 3.2 billion letters.
In a human genome.
In a human genome.
Yeah.
Yeah. So it's just a long list of those. And so that's just your DNA, that's your genetic material. So every time your cell replicates, it copies itself, it copies that DNA.
Yeah.
And makes a new copy of that. And that DNA contains all the information needed to build you as a person. So the DNA, maybe like the thing that does the most we can talk about is that some parts of the DNA is what we call genes. Like this long stretch of bases. Let me just show you an example of what that could look like. Like here. So this is a little
gene.
This is a genome. A little part of this is the mitochondrial genome for a Neanderthal. And you see, it's just, I don't know if you can pick that up, guys. Just a long list of letters. But then some parts of this region of the mitochondria has genes. So let's just make it up an example. Like say this region here is a gene. So what happens is that this DNA gets turned into proteins. Mm-hmm. And these proteins are then the things that, for instance, like that you need to build your cells. Like so your nails are made of protein, and the little things in your body that breaks down food, for instance, are proteins. And a lot of your cells, like a lot of the things they're made of, is also protein. So that's when if you want to grow, for instance, like a lot of bodybuilders eat a lot of protein. Like that's like building blocks for your body.
Yeah.
So that's what they do. So the DNA gets turned into proteins that does something, and that region of the DNA that gets turned into a protein we call a gene.
Okay. So the region of a genome gets turned into a protein is a gene.
Mm-hmm.
Okay.
Exactly. And then, in the human body, there's something like 20,000 genes or something.
Yeah.
And they all interact with each other. And they all do things. And so there's also different versions of genes.
Yeah.
And that comes from the fact that, so this little DNA sequence here, that I'm highlighting here, could get turned into a protein. But if every time your cell copy itself, it has to copy all this DNA. And sometimes it makes mistakes. Mm-hmm. So it could, for instance, make a mistake where it turns this G here into an A. So that would be like a mistake.
Yeah.
And sometimes that has an effect. Like if that base change, the protein that it encodes can change. Mm-hmm. So maybe I should actually pull that up real quickly.
Do the C and the T and the A and the G, do they always go together in a certain combination?
Yeah.
What is that?
So the DNA is like double-stranded.
Yeah.
And so what happens is that the base pairs chemically bond with another strand that's called the reverse strand, and they always pair the same way.
Okay.
So a C and a G will pair, an A and a T will pair.
Okay.
And the reason they're smart is like when the cell is actually replicating, what it's doing is it's breaking apart the DNA strand, and because they are both identical, they always pair the same way. It can just fit in all the molecules on this one, mm-hmm. and because they always pair the same way, it can do the same on the other one. Yeah. And then you have two strands.
Okay. I think I'm following.
Yeah.
It's crazy that this is going on in my body all the time.
Oh, yes. Yeah.
Like, I presume every time a cell replicates.
It's also replicating all the time. And in some sense, I think it's like 7 years from now, there won't be a single cell in your body that's the same cell that it is now.
Yeah.
I think skin cells are quicker, like they die quicker than other cells.
Yeah.
So in some sense, you are like it's a thesis-ship kind of thing. Like you are like a entirely new person.
And yeah. It's like that question where it's like, would you go through a teleporter if it like erased your body and just rebuilt it perfectly?
Oh yeah.
At another thing. Yeah. But that's kind of happening slowly all the time
really
anyway. Which way? How do they pair again?
C and G and T and A.
C and G and T and A. And at each sort of point in that helix is a base pair. Is that right?
Yes, exactly.
And we have 3 billion base pairs in our genome.
Yes.
Okay. That's our DNA, guys. I didn't even realize that a gene was specifically a part of your genome that made a protein.
Yeah.
Like, I just thought it was just, I don't know, the part of your genome that you're talking about. I didn't even realize that had a specific
thing. It is just the DNA
thing.
being turned into to a protein, like a long series of amino acids. And then I wanted to show you how we get like the process for all the way from the beginning, like when we get the DNA, what format it is in, and how we convert that into what is the DNA sequence of this Neanderthal or human.
Yeah. That's interesting, yeah.
So here's a little sample of like a Neanderthal genome.
Yeah.
And you know DNA is double-stranded, but I'm just showing you one strand here. Because the other strand is the same information, basically.
Yeah, because if they always
Because they always pair the same way.
Yeah.
So you typically just show one of them.
Okay.
Because the other is redundant information. So this is a little Neanderthal genome here.
Yeah.
So ideally, what you would do is that you would just like take your ancient bone or whatever that's Neanderthal and have a little machine that can just read this. Mm-hmm. Like from beginning to end. And then we have the full DNA sequence of a Neanderthal. Mm-hmm. But unfortunately, you can't do that.
No?
No, because Neanderthal DNA, it's old, first of all. It's like thousands of years old.
Yeah.
And as with most things just fall apart. Mm-hmm. So it gets, like all the DNA that's in the bone gets broken down. And there's also some particular type of damage that happens over time that I'll show you in a little bit.
Yeah.
So it gets broken down into these small pieces. So actually what we end up having is we get these little pieces, like these small sequences of the DNA that we can actually read. So this is what comes out of the sequencing machine. But okay, so then we have like a list of these. So what do we do with them now? And then what you do is like, you gotta find out where they, so you're comparing to something always. So we're comparing to the human genome, for instance. Yeah. So we're looking for differences between the Neanderthal and the human. And then what you do is like you sit and you figure out where it fits.
Okay.
Here. So this little bit here fit over here. And let's take this one fits here, right?
Okay, yeah.
Yeah. And then this one fits here, and then this one, let's see, here, right? And then, so then you sit and like you do this little, well, you don't do the puzzle. The computer.
You're not dragging them.
Yeah. No. It's
3 billion times.
It's a lot of data. Like when I was working on there's like a Neanderthal from Vindia Cave in Croatia.
Yeah.
And when we got the data for that, we got these little reads here. And all that data, it was sent on a hard drive because that was quicker than transferring it on the internet back then. So it's like this actual hard drive sent around to people. And that was, I think, a 2 terabyte hard drive filled up with these little reads here.
Wow.
That you then have to sit and fit.
And the machine is trying to fit them in.
Exactly.
To a reference, like you'll have a Neanderthal reference genome.
Yes. Or you use the human genome, or you use the chimpanzee genome or something.
Okay.
All you want to do is like figure out how similar is it.
So you use like a human and chimpanzee genome in understanding.
Yeah, exactly. Most of the time, the human one.
Okay. I don't know if this is an easy question to answer or not, but how many differences in these 3 billion letters are there between like a human and a Neanderthal and a chimpanzee?
Millions and millions.
Millions.
It's something like between humans and Neanderthal, I think it's one difference every 1,300 base pairs or something like that.
Well, one difference every 1,300 base pairs. When you say it like that, it sounds like we're really, 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 really similar.
We are really, really similar.
Yeah.
Like, it is like really like most of the genome is very similar. Like between you and me, like maybe out of the 3.2 billion, it's a couple of million differences. The rest is the same.
Wow. That's kind of crazy.
Yeah, and then chimpanzees maybe 10 times more different.
Yeah.
So it's like one difference every 120 base pairs or something like that.
120,000 base
20.
pairs?
Like one difference out of 120.
Per
120.
One difference per 120 between me and a chimpanzee.
Something like that. Yeah.
That also feels like not a lot.
No.
One difference every 120 base pairs between me and a chimpanzee. That's kind of crazy.
It's not so much. Let me show you actually what.
That's what people mean when they say a human and a chimpanzee have like 99 point whatever percent the same DNA.
Sometimes it's like sometimes people report the entire genome, and sometimes they only report genes. So then it's a bit different because genes they're not free to change so much because of natural selection. If a gene does very important changes too much and it breaks, that DNA is not passed on because the individual dies because it was so important, this thing.
Like
so
a gene associated with, say, breathing or something.
Yes.
For example, probably not much variation.
Yes, and like all the like the things going on in your body that takes if you eat food and converts that into energy, like those things. If they change, then you are screwed.
Yeah.
But
probably they haven't, like, not just between us and a chimpanzee, but those genes are probably shared between like all sorts of life.
We go back, like we share genes with all the way back, like trees, fish, like, there's some really ancient genes. Like they change slightly, but the function like remains the same.
Yeah.
But let me show you here. So now I'm gonna show you. This is the Chigis-Guy. Remember those from Chigis-Guy cave? So this is just one individual, and this is the data that we get out of it. So this is the data that comes out of the machine.
Yeah.
And you can see there's a lot of numbers, but these ones you are familiar with. So this is just all the small reeds I was showing you before. This is just, they all have names, and they have like unique names. So that's just what these ones are. But all these ones are the actual pieces of DNA that we need to fit together in this puzzle, that we need to align.
Okay. So you'll get just like, what are these different positions? I mean, or just different chunks of DNA that
different
it's
chunks, and we don't know where they fit.
Okay.
And now the goal is like to do that little exercise we did and make them all fit.
Okay. So after the genome's been sequenced, you just get a list of chunks of genome.
Yes.
And you have no idea where they should go at that point.
And there's something like how long are these? Like maybe 30 to 50 bases or something like that.
Yeah. Something like that.
If you sequence a modern human genome, you can get like things that are mega bases or thousands of bases long. But because this is ancient DNA is broken down. Like over time. Like
okay, yeah.
The DNA like your body degrades, and the DNA also degrades.
Yeah.
And then let me show you what it looks like. So this is a program that can visualize all these reads and how they.
Okay. Now something's just clicked in my head because I didn't fully appreciate how similar our genome was to a Neanderthal's genome. That's how you can kind of try and find the place where they'll go.
Yes, exactly. Because if they're too different, then you couldn't figure out where they went. But because they are so similar, like we share a common ancestor, like we're all great apes in some sense. Like you can like find where it aligns, it's called, and then look at the differences. And I actually have a little example here.
Now I understand. So this whole thing really relies on just how similar we are, in a way.
Yes.
Okay. We're learning together, guys.
So these are all the... Let me just color it by the strand. So this is like all the reads that are mapping. So like there's one here, for instance. And it only shows you where there's differences.
Yeah.
So every time there's not, like some letter is the same. And then you could maybe say like, so you see like there's a lot of like reads overlapping. The number of reads per position, I think you talked about this in one of the videos, it's called the coverage.
Yeah,
that's when we talk about high coverage, low coverage.
Yeah, my last mammoth video, yeah.
Exactly, yeah. So this one has a coverage of 2,600, but this is mitochondria.
Yeah.
And you have hundreds of copies of mitochondria in your cell, so that's why it's so high. The nuclear genome, there's only one copy. So the coverage for that is way lower.
You have hundreds of copies of mitochondria?
Yeah.
I thought...
Little powerhouses, making all your energy. Your muscles have more, because they need to make more energy. Yeah.
I had no idea. I'm learning a lot here, guys. I don't know why. I thought there was one.
No.
I just thought there was one. I don't know why.
So then you can see all these reads. And there's another thing here. Have you noticed that all the reads that are like colored red have a lot of T's? And all the reads that are colored blue have like a lot of A's. Some of them also have T's.
Yeah.
That's ancient DNA damage. Because over time, DNA gets shorter.
Yeah.
But there's also something specific that happens to it. And that thing that happens is a chemical process called deamination. But it essentially turns C's into T's. And it turns G's into A's. But it only turns C's into T's on the forward strand. you know that there was 2 strands.
Yeah.
We call one of them forward and one of them reverse.
Okay.
So they will be colored in red, the forward, and the reverse are in blue. So these.
So this is like either side of that double helix.
Yes.
Okay.
And this thing is what we use to know that this is in fact Neanderthal DNA, because this only happens with old DNA. So if let's say an archaeologist, I don't know if you know this, but like they bite the bones to figure out if it's like they lick the bone to figure out if it's a rock or a bone.
Yeah.
That's gonna contaminate it. That's gonna introduce human DNA. But that human DNA is new, so it doesn't have this damage pattern, this ancient DNA damage pattern. And that's how we know it's real ancient DNA.
Interesting. I mean, hopefully the days of licking bones are over now that we have ancient DNA. Hopefully that is...
They're not.
At least a human bone.
Yeah.
Hopefully we're not licking them so much anymore.
But then another thing here is like you can see like there's all these damages, but there's no changes to that sequence that we're comparing it to. And that's going to become apparent like in a little bit. So the mitochondria is something like, that's the smallest part. That genome in the mitochondria is only 16,000 base pairs. So that's actually very short. Like that was the one I was showing you here. You can scroll through it. So as we're scrolling through this, that's the same as we are scrolling through this. So you see some, maybe there's like a difference here. But I wanted to show you.
And each line of these is a different read of the DNA.
Yes.
What do we mean by a read of the DNA?
Oh, sorry. So like each line, each line here.
Yeah.
It's like a little read. So that's this. So this visualized would be like this thing here, for instance.
Okay.
And then each column, it's like the first column would correspond to this position 1 in the mitochondria, and then position 2 and position 3 and so on.
Okay.
That's what this is. But I wanted to show you, there was an example I had where there is actually a difference. And I think I wrote it down because I know I couldn't find it again. Here, can you see this?
Yeah.
So here there's an actual difference between the DNA of the organism that we're looking at, which was Tekiskaya A, and the reference genome we're comparing to.
Okay. So
you can see like
there's a G. Yeah. In the Neanderthal there's a G, and in a modern human there would be...
Yeah, it's actually another Neanderthal too, but...
another
Neanderthal?
I'm
comparing two Neanderthals.
Okay.
So that's why there's only one difference. So here there's a difference. So what you could do now, like, you know that the mitochondria is just a sequence of DNA. You can just read off the sequence, so you can say, it's most of the time it's the same as the reference, except for here. Here's different. So you just print out a sequence that has the same letters as the reference, except for where it's different. Then you print out that difference.
Okay.
And those things, so then we have a DNA profile of the individual we were looking at.
Yeah.
And those things you can now compare to, for instance, the DNA sequence of a human, the DNA sequence of an orangutan, or a chimpanzee, or something like that. So let me just do that. And I have another program for that. And then let's take this file here. It has like 54 humans, some ancient humans, some Neanderthals, some Denisovans, an individual called Sima de los Huesos, which is also a Neanderthal, and a chimpanzee. And let's just analyze that. So this is like all the different individuals. You maybe know like Mesmaskaya 1 is a Neanderthal, Stantofa 1, 2, Vindia Neanderthals, Esidron Neanderthal, Altai Neanderthal, Klaknikov Neanderthal. So this is like all their sequences.
Yeah.
And if there's a dot, it's just the same as the reference. And now I can color all the places where they're different. So here, for instance, there are some individuals that have a difference.
Yeah.
And here, there are some other individuals who have a difference. And then the idea is that because everyone shares a common ancestor, things that are very similar should be closer related. They should have a more recent common ancestor. And things that are more different have a more distant common ancestor. So we can just count all the differences between them. And then using this, let me show you something really cool. You can make a tree out of it.
How can you tell the tree just from the differences?
So that's just the idea that like if two sequences have like one difference between them, and another sequence has 10 differences to both of those sequences, these two must group together first.
Okay.
And then the other one will group in. So let's try to make a tree and see if we can make it make sense.
Yeah.
So now I'm just making a tree. So what do we see in this tree? We see that chimpanzee has a very distant, and this is like the common ancestor is here.
Yeah.
And then sort of time passes on here.
Mm-hmm.
So chimpanzee that's an outgroup that has like a very distant common ancestor with Denisovans, Neanderthals, and modern humans.
Mm.
And then you see here in the tree that, okay, then Denisovans are actually like an outgroup to humans and Neanderthals.
Mm-hmm.
This is the mitochondria only.
Yeah.
And here we have all the humans. And that's how you make a tree, and that's how you say, this is how humans and Neanderthals and Denisovans are related.
My brain's working really hard here. So hang on, let me see if... So we make the tree by sort of counting the differences.
Exactly.
And so if, I don't know, just for the ease of number, a chimpanzee has 100 differences at a point.
Yeah.
And us and Neanderthals have 10 differences at this point.
Yeah.
Then we must group together separate from the chimpanzee.
Exactly.
So it's just counting the differences.
It's just counting the differences. Then I think we can, so these are all modern humans. I think we can compress them. Maybe that's a bit easier to visualize.
So that triangle is all modern humans.
That's all
now.
modern humans, yeah.
Yeah.
And then there's some Neanderthals here, and I think we can. Yes.
That's interesting that the Czechoslovakia Neanderthals group closer to like Western European Neanderthals than other Eastern European Neanderthals like Mesmaiskaya.
Exactly. And here, somewhere here, you will also have the Altai Neanderthal, also from Denisova cave.
Yeah.
But it doesn't group with them. And that's actually something like archaeologists figured out first, because they looked at the stone tools. Mm. And the stone tools in Denisova cave, look very different from the ones in Czechoslovakia cave. Mm. And the ones in Czechoslovakia cave look more like stone tools seen in the Caucasus and in Europe.
Yeah.
So they actually hypothesized that the Neanderthals in Czechoslovakia are some migrants from Europe that came over there. Mm. And then when we look at the DNA, we see the same thing.
Yeah, interesting. I did see as well that there had been a debate for a long time. There seemed to be like a subtle difference in Neanderthal anatomy for a little bit, and they wondered if it was like some group had expanded and another group had contracted and stuff, and then like DNA did later confirm that.
Yeah, exactly. Yeah.
There's probably this same situation here.
Yeah. So that's really cool. And then you can also see, like, have you ever done a video where like humans and Neanderthals, their mitochondria is more similar? Why that is?
No.
Like, so there's this hypothesis that Neanderthals, their mitochondria and their Y chromosome is actually human in origin, and it's been replaced.
I had heard that, yeah.
And that's why you see this thing, because this one here, I don't know if you can zoom in. That's Simandidus versus, and that's a Neanderthal.
Oh, yeah.
And that's something like 400,000 years old. And then after that, some human population leaves Africa, meets the Neanderthals, and gives them a little bit of human DNA, but it completely replaces their mitochondria and Y chromosome. Yeah. So that's much more similar between Neanderthals and humans. And that's sort of what you're seeing here.
So this mitochondrial DNA of Neanderthals is closer to a modern human than it is to a Neanderthal that lived 400,000 years ago.
Yes. But that was just the mitochondria. The genome is way bigger, and there's way many more connections in the genome. So genome-wise, Neanderthals and Denisovans are more like each other. Mm-hmm. But mitochondria and Y chromosome-wise, humans and Neanderthals are more similar to each other.
Interesting. If we're just counting the differences, how does it know that our ancestor with chimpanzees is much further back in time? Is it just because it takes X amount of time for that many differences to accumulate?
Yes. So the thing about these differences, like, they happen every time your cell replicates. Yeah. There's a chance that there's like a mutation, it's called.
Yeah.
And they actually, so you can only pass on mutations if they happen in your sperm cells or your egg cells.
Yeah.
So they will be passed on to your child. Mm. And on average, number of mutations, it's something like each individual has 60 new mutations in their entire genome. Mm-hmm. And that depends on the parental age. Yeah. And especially on the father's age. Yeah. Because sperm cells regenerate more.
Okay. I don't know if this is a crazy thing to say or not, but because the fathers are accumulating more mutations because our sperm is just, constantly in production. Does that mean that like a lot of changes in the genome, most of them come from the father's side?
Yes.
Interesting.
Like 75% of mutations come from your father or something.
Oh, wow. So all of these things, all we're doing is counting the differences. And if there's a lot of differences, that must have taken time to accumulate.
Exactly.
And you can tell that things are closely related because they have less differences.
Exactly.
So you would know, like, let's say you were given a genome of an oak tree, a penguin, and a Neanderthal, and us. You could group all of those just from counting the differences.
Theoretically, yes. It gets a little harder because, we have 23 chromosome pairs.
Yeah.
Like great apes have 24 pairs, for instance. Yeah. Like some of the chromosome gets deleted or duplicated or merged or something. So it gets harder and harder to find places that actually this is the same piece, just with a lot of mutations. Because at some point it becomes so different that we can't align anymore.
Okay.
We can't accurately map them.
Okay, so there's not like not many pieces of our genome.
Yes, like there are all the genes. Like they are more constrained.
Yeah.
Yeah.
So these mutations going on CG80, and let's say there's a mutation in one spot, it becomes a T. Like, I don't know. Let's say, how can I explain it? Let's say it becomes a T here. All of these guys would have a T in that point, but these guys will have extra mutations.
Exactly.
And these guys won't have the T.
Yeah.
Because the T happened after this split between us and them.
Exactly. So what we do actually a lot of times is
my brain is struggling, guys. Like a crash course in genetics.
We do something called like the molecular clock, where you try to figure out how old the sample is.
Yeah, I was going to ask about
that.
Like
you just count how many differences is there between this sample and a chimpanzee. Because the differences, if you think mutations happen at a certain time, must be like due to how much time you've been alive compared to chimpanzee. So like a modern day human will have more differences to chimpanzee than a 45,000-year-old human.
Because we've had 45,000 more years to pick up mutations.
Exactly. And that's actually what they did with Usishim, which is like an ancient human found in Russia. Yes. And they did that, and they counted how many missing mutations, and they estimate that the individual is probably around 45,000 years old.
Yeah.
And that's also the radiocarbon date.
Yeah.
So it sort of works.
So okay, interesting. I wondered about that. So when you're doing the molecular clock, you're just dating how many mutations difference between us and a chimpanzee.
Exactly.
And because we've lived longer than someone that lived, like you said, 50,000 years ago, we've got more mutations than they did between us and a chimpanzee.
Exactly.
And it's such a relatively consistent rate. What's the plus and minus here? Like,
let me maybe like this is a mitochondria, so it's only 16,000. So there's not so much information, so you get like pretty wild estimates.
Yeah.
I remember for the Chagiska ones, it's something like they lived between 40 and 90,000 years ago.
Yeah.
So plus minus
40 and 90,000 years.
That's 50,000 years of like difference, right?
Okay.
Like we can say they're definitely not like modern. They don't just die. They didn't just die recently.
Yeah.
It's definitely a while ago. But the time is a bit hard. So that's for the mitochondria. You can do a bit better with the nuclear just because it's so much bigger. There's more data.
Yeah.
So you can get more precise estimates.
What would you say is with the nuclear DNA, the most precise genetic dating can be? Are you confident you would get within 10,000 years of when that person lived?
No. And that's more like it all has to do with when did the recent common ancestor live.
Yeah.
But that process has a lot of variation in it.
Yeah.
So it's never gonna, radiocarbon is always gonna be better. That's gonna be in a few hundred years. This genetic thing is always gonna be in thousands of years.
Okay.
Unless the size of the genome went to infinity, then you would get pretty good.
Okay.
But that's not, our genomes are not getting bigger.
But what is it like? It's always like 20,000 years. You think nuclear DNA? What's the?
We could say that plus minus a couple of thousand years, 10, 20,000 years. Yeah.
Plus or minus 10 or 20,000 years.
That's probably something like that.
Can I just ask though? I've heard about geneticists calibrating the genetic clock with like ancient DNA. Is that almost like, oh, this individual, say, I don't know, 15,000 years ago had this mutation, so we know it happened by then?
Yes, it's something like that.
Yeah.
It's using radiocarbon data to try to say, because there's so much uncertainty in the genetic ages, trying to say, okay, but this individual, we know from radiocarbon data, that's 40,000. Okay, then we can maybe use that to...
Because you can see what mutations they had, and then it'll be like, oh, well, it appeared by this point.
Sort of like an anchor point.
I don't know if I'm jumping ahead here, but I know at the minute we're talking about species that we have reference genomes for.
Yeah.
And I know you're trying to work on a project to find admixture where we don't have a reference genome, I suppose.
Yes.
So, how?
How's that going?
How can we even do that? If we don't have the reference.
I have a nice little example here, actually.
For those of you that don't know, Laurits is going to start working on a project soon to try and find out if there's any more episodes of like intermixing between us and other archaic groups
that we don't have ancient DNA from.
That we don't have ancient DNA from. You know, kind of like how we discovered the Denisovans, right, when we found that thumb.
Yeah.
Maybe there are more groups out there.
Yeah. I actually have a little example here. You can feel free to use these or not.
Yeah.
So I like to say that we live in very extraordinary times in the sense that we are the only types of humans alive right now.
Yeah.
But if you go back 100,000 years, you know there's at least 7 types of humans. So I have like modern humans here in Africa. That's where we presumably were.
Yeah.
Neanderthals in West Eurasia, colored in blue, and Denisovans in East Eurasia, colored in orange. But then there's all these different humans that we don't have DNA from yet.
Yeah.
We have the remains, but we don't know how they connect to us in a tree.
Like Homo erectus.
Exactly. Yeah, and I showed like where do we have ancient DNA, like high coverage, good quality genomes. And that's just 3 Neanderthals and a Denisovan.
Yeah.
But let me show you how I'm planning to do it. So the way I like to explain that is thinking of DNA as languages and thinking of a little piece of, let's say, Neanderthal or Denisovania or something else as a borrow word from another language that comes into our language. So I have a little example here where I have some Scandinavian languages, or some Nordic languages I should say, and then English. And what you can maybe appreciate is that Danish uses the English word for weekend. Like we use that as a borrow word. So in the case where we have Neanderthal genomes, then you would just say, and so this is like the language analogy, and then over here is the genetic, like what the genetics actually is. So we have different people here with their DNA sequence, and I highlighted like when there's differences between them.
Yeah.
So this little person 4 here has some Neanderthal DNA that came in here. That's the borrow word. So if you had Neanderthal genome, you can say, here, you're more similar to the Neanderthal than you are to humans.
Yeah.
So that's easy peasy. But now we're going into the example of what if you don't have any Neanderthal DNA? So I'm gonna take this out now.
Yeah.
What do you do then? Well, even if you didn't, in languages for instance, even if you didn't know English existed as a language, you would still maybe appreciate that the word weekend is very different from the Nordic word like helg or helgi.
Yeah.
And that's what's driving this signal.
So you're just looking for like very diverged parts of the genome, I suppose.
Yes.
Like where there's an obvious difference between,
let's say, like I looked at these for like some people from Europe or whatever, and one person has a very different little piece of DNA here.
Yeah.
Then I would say, I think this is some archaic DNA. And I tested it out, like where I ran this on a bunch of modern human genomes, and you find a lot that my program then says this is probably like a little piece of archaic DNA. And then after I found it, I can compare it to Neanderthals and the Denisovans just to check that it's actually working. And it does find a lot of things that are more Neanderthal-like and Denisovan-like.
But not from Neanderthals and Denisovans, or...
I'm suggesting that it works. Like,
I see.
They are from Neanderthals and Denisovans.
So you're running a program without telling it like Neanderthal and Denisovan DNA.
Exactly. It doesn't know that exists.
Yeah. And it identifies these points like, hey, these points are highly diverged or whatever.
Exactly.
And then when you look,
then they actually turn out to be very similar to the Denisova or Neanderthal.
Okay. Yeah. So it's just trying to find. So how would you know that a part of the genome is that without this reference from archaic DNA and not just a mutation that you have? How would you?
Yeah. So here we actually have to go a bit back to the tree building.
Yeah.
So I am gonna use the Neanderthal and Denisova, and I'm gonna take all these little pieces that I think are archaic, and I'm gonna make a whole bunch of trees out of them.
Mm-hmm.
And then you're actually gonna see. Let's say, let's go back to this one here. Let's say these are all. This is like just an example. These are all the humans now. And then I find a little piece, and that piece fits here in the tree.
Mm-hmm.
Then I can say, oh, this little piece is this mysterious hominin, but it's very closely related to the Denisovans. So it's probably a Denisova-like population.
Yeah. So, but how do you know it comes from that chunk, though? That's what I still don't understand. Like, you have like this DNA that comes from that. How would you know it came from like this archaic chunk?
So I have, like, let's say I am using the Denisovans and Neanderthals, but I have this little mystery chunk.
Yeah.
And then I take this little mystery chunk and I make a tree comparing that to Neanderthals and Denisovans, like all the same principles that we talked about before. And then maybe the chunk falls here, and you say it's more Neanderthal, or it falls like completely outside all the variation that we see in humans, Neanderthals, and Denisovans. Then maybe it's a Homo erectus kind of chunk.
Okay. I'm trying to think if I understand. So you'll compare it to humans and Neanderthals and Denisovans, and
then we're gonna see how many like variants that are unique to this chunk, for instance. That if it had a lot of unique, it's gonna be like outside of the variation.
Oh, I see. Yeah, because we're counting the differences, I suppose.
Exactly.
Yeah,
it's all counting differences.
So if there's more than you would expect, then it's going to fall outside of the range of Denisovans and Neanderthals.
Exactly. It's going to be some Homo erectus or whatever.
Because we're more diverged from Homo erectus than we are from Neanderthals, so you'd expect there to be more differences.
Yeah, that's the...
Guys, it's just counting differences. Like,
like all like biology is just comparing two pieces of DNA and seeing how different they are.
Yeah. Okay. What do you think your chances are of finding other hominin groups?
I mean, I think, if I'm being optimistic, I think they're pretty good actually.
Yeah.
Like, so we know Neanderthals existed. We see all the remains of them. We get some DNA out of them, and that DNA also happens to be in humans. Like, we met Neanderthals and we had children with them. We found a little finger bone from a Denisova.
Yeah.
We sequenced it. That there's also Denisovans in modern humans.
Yeah.
And it seems very unlikely to me that the two ancient humans we got DNA from just happened to be the only two that contributed doing DNA to us.
Yeah, and what would be the chances of that?
Yes, and with all the humans around, we didn't know Denisovans existed until like 10, 20 years ago or something like that.
Yeah.
I think there's more humans out there, and I think our ancestors met with them, and we have some of the DNA.
Yeah.
So, but now I'm just trying to quantify how much is it, and is it really like is it a completely new type of human, or is it actually maybe something that's as close to us as Neanderthals and Denisovans are, just a sister group to those? You know what I mean?
Yeah.
Or is it like something like Homo erectus, or maybe even way more ancient? So actually, like, if you just count differences, there's actually, it's almost the same if you compare two humans and a human and a Neanderthal.
Yeah.
It's the type of differences, so it's where they fall in the tree, if that makes sense.
Yeah.
And again, when we're talking about the nuclear DNA, it's an average of the entire genome.
Yeah.
So just on average, Neanderthals has a bit more differences. But many times you really can't tell. You can only tell a bit like when you
see the differences.
Yeah, when you're doing this comparison. Like, there's some model that is like taking this information and making it into, I think from here to here you are more like Neanderthal.
Yeah.
I don't know. You did the 23andMe, right?
Yeah.
Yeah. Remember, like in the genome, when it colors the genome, says like you're more like French here. You're more.
Yeah.
That's the same thing this is doing. It's like it's still it's human, all of it, right? Like, and you are very closely related to all the different populations. Just like here, it thinks the best match is French.
I just share,
share a little more. You know,
a
you
little
still
more
share
with the French
most.
guy. Yeah. I've been actually wondering about a video about this because I see on like TikTok and stuff Asian people being really unhappy with 23andMe because it always gives them the result 100% Chinese.
Oh.
and when you do a European, it says
Yeah.
like you're this bit French and you're this bit Norwegian and blah blah blah.
And
blah,
this particular town in Ireland or whatever. Yeah.
But I wonder, is that just like you could also like give my result back and say you're 100% European.
You're good. Yeah.
Like, is this just how we're like classifying things? Like,
it's just like it's the database. Like their database, like their customers are mostly European, so you have more resolution to say where in Europe is it.
Yeah.
And if their customers was mostly from China, you would be able to do that same thing.
Yeah. But I suppose what I was wondering is it like it's just an amount of classification. Like Europe is not a united country, so it says like, oh, you're all these different little groups, whereas China is a united country, so it just says you're Chinese. But who has the most variation? Like, is someone from the very south of China and the very north of China, do they have more genetic variation than like a British guy and I don't know, a Polish guy? Like, who has the more actual genetic variation?
Yeah, I know what you mean.
Maybe we're just classifying it in a strange way.
That's I think that's it, because most non-Africans are very, very similar. Like,
most who?
Non-Africans.
Yeah.
Almost all the genetic diversity exists in Africa.
Yeah.
And when our ancestors left, and like when people left Africa and that became the ancestors of all non-Africans, it was like a little group that left. Yeah. So we trace all the DNA back to those. So there's not a lot of differences.
Yeah.
Between non-Africans.
That's what I was thinking. Yeah, like I know that's a massive deviation from what we were talking about.
Oh, yeah, that's something else. Yeah,
I've just been wondering in my head, you know, I've just been thinking about DNA so much these days.
Yeah.
Thinking of questions. Is there any limit to how far back you can look with these kinds of things? Like, let's say, I don't know, we had an admixture event with an unknown archaic group 170,000 years ago.
Yeah.
Would that be hard to detect as compared to like
it wouldn't be
55,000?
Yeah, it would be harder. And it would be harder because the signal, like these little blocks of DNA, become shorter and shorter and shorter.
Because they get recombined every generation.
Exactly. Yeah. And the shorter that, like if one of my parents were Homo erectus, that would be very easy to detect in the genome. But if it happens like, 100,000, 200,000, 300,000 years ago, it gets harder and harder.
It's just a part of, and
the signal just
when it goes so
down.
far back, like it's within absolutely everyone, I suppose, as well. Like there's no variation probably.
Exactly.
Between humans, whereas I imagine you're trying to find groups that we admixed with roughly as out of Africa was going on.
Something like that, yeah.
In that sort of like 50 to 100,000 years.
In the past 50 to 100,000 years, yeah, exactly.
Where you think, is there any like human population that you think would have the most chance of identifying these signals? Because obviously, like you said, we have a lot of genomes from Europe, and Europe is where Neanderthals were.
Yeah.
And these skulls that are hard to classify tend to be in East Asia.
Yeah. Like it would be really cool if people who donated the DNA wanted to figure out like if there were some archaic groups here. I think it could be really interesting to get a lot of genomes from South Asia and East Asia and this region here, because that seems to be where a lot of like different types of human groups were living.
Yeah.
Whereas like up in Europe, it's mostly Neanderthals that we know of, at least.
Yeah.
That have been around.
Because I think we've already identified that humans admixed with two different groups of Denisovans.
Yes.
I think. I mean, maybe even more, but...
At least two.
At least two, yeah.
And that's also the thing, like the Denisova genome we have, I think I have it here, is up here from Siberia.
Yeah.
But the Denisova population we met was probably not up here. It was probably maybe more down here.
Yeah, I did a video with Joam Teixeira, a geneticist, and he thinks that fossils that we've identified as Homo erectus in like Java, Indonesia, he thinks they could very well be Denisovans. And he was also talking about potential Denisovan admixture within like Indian populations and stuff that, you know, they feel like they've detected.
Yeah,
like,
I think it's definitely here where a lot of cool things are happening. But then again, like look at where these like remains are found. Like there's also like a huge part that we don't know so much about.
Yeah.
Like here in Africa.
Would it be easier or harder to detect like admixture in Africa? Because Africa has sort of been this place where we've been living for a very, very long time.
Yeah.
You know, since we split from chimpanzees.
Yeah.
There's been humans evolving in Africa. So does that make it harder to detect different groups in that region, because these different groups have all been contributing to our evolution for a very, very long time?
Yeah.
Whereas we have out of Africa, like you said, if your mum was Homo erectus, it would be this like really clear signal.
Yeah. And also because it is really hard, let me just say it like that. Yeah. This is by far the hardest place. And it's also hard because there's not a lot of genetic diversity outside of Africa. So it's easy to do this piece of DNA looks very different from these other pieces of European DNA, say, so it's probably a Neanderthal.
Yeah.
But because there's so much genetic diversity in Africa, it gets harder. And also it gets harder because we don't really know how we humans came about. Like, is it one group or is there multiple human groups all over Africa that like, is it like a network of groups exchanging migrants with each other?
Yeah.
Or what's actually going on here? And at one point, like, if a group is, say, 500,000 years, that's like human Neanderthal. Like if their common ancestor lived 500,000 years ago, oh, is that a different type of group? What if it's just 400,000 or 300,000 or 100,000?
Yeah.
You know, it's
that's what I wanted to. That's what I wanted to ask you as well.
When is it different enough that we say this is archaic integration, and when is it just two populations meeting?
Two populations.
Two human populations.
Yeah. So what is your? That's what I wanted to ask you. That's a great segue. What is your, like, when you think of our species? You know, for a long time, Neanderthals, Homo sapiens, we're different species. We have this different biological classification. How do you view all of that as a geneticist? Like,
I think the species concept like goes a bit out of the window here because like we clearly have Neanderthal distributed DNA. So in that sense, they're not different species. Yeah. Because we could have like offspring clearly that survived because like we have the DNA today.
Yeah.
So it is this kind of weird thing where like we're trying to put something non-continuous on something that is actually continuous. And what I mean is like this species is something we made up. It's a classification we made up. But like it has to be something like there's all there is just DNA. There's just differences.
Yeah.
And at one point those differences are so much that you cannot produce offspring anymore. And then maybe you can say you're different. But there's like a gradient. It's a sliding scale.
Yeah.
Yeah.
Like, to use that example of Africa, you know, there are all these groups in Africa that probably were coming in contact more frequently than Neanderthals and Denisovans and this African population, just because of geography.
Yeah.
But really, when you zoom out, it's almost like we're all human species, and some we're coming into contact with very frequently, and some we're coming into contact with very, very infrequently.
Yes, I think that's the right way to think about it.
But to say like we're one group and they're another group seems so ridiculous almost. I don't know. It's just
no. I completely agree. Like it is just all these like populations meeting, being apart, meeting again, being apart. Yeah. And at some point, like they're apart long enough that we say now they're two different groups. Like also like why is Denisovans and Neanderthals like two different groups? Like they could also just be a human group, and why are we not also part of that human group? Like it is just.
I mean, we are. I kind of feel like we are. Yeah.
Yeah, like I mean, like their DNA lives in us, so it is human DNA.
Yeah, and we kind of think like, you know, because they were separate for a long time, and then modern Homo sapiens expanded out of Africa, and for the most part, for whatever reason, we've replaced them. Like, similar dynamics could have been going on in Africa, right? Like, one population, for whatever reason, at one time, expands massively. Mixes with another group, and then at another time, another population expands massively.
And that's what we see with the Neanderthals too. Like the European Neanderthals or the one from the Caucasus, like the Cherkesskayas, like who are over here, are more like European Neanderthals than they are like the Altai Neanderthal, for instance. Yeah. So this probably happens like all the time.
Yeah.
Like, groups move around and some expand, some contract, and some get absorbed into another group, and yeah, it's complicated.
It's kind of complicated. It's interesting though.
It really is.
It's interesting how much like ancient DNA has changed our view of like what even is a human. Like I did a live stream the other day, and like now I find it so hard to say anything concrete about humanity or evolution or anything. It's like it's like the more I know, it's like I don't want to say anything concrete about how many archaic groups did we mix with? I don't know.
A few. Well, maybe like maybe they're just like different human groups that we met and mixed with. Yeah.
And it's like, is it out of Africa? Is a human evolution like out of Africa or multi-regionalism? It's like, well, kind of both. Now, it depends on degrees. It depends on what you're placing the emphasis on. It's so hard. Like you said, this is like a continuous biological, organic process.
Exactly.
And we're trying to put like such neat little boxes
exactly.
around it.
Yeah. And like the early models, a lot of like human evolution models is like, we have a common ancestor with Neanderthals and Nisovans, they split off, they never meet each other again. They never exchange like anything. But that's of course not how it is. Like they keep on merging.
Yeah.
I heard someone describe it as like instead of, you have a river that splits off, or like two rivers that join.
Yeah.
You have like a muddy delta of something.
Yeah.
Of a lot of, like, it's something like that. It's more like what's actually happening.
Yeah, like a river delta. Like some branches might contribute a lot.
Yeah.
And some might contribute very, very little, but they're still interconnected in so many like intricate subtle ways.
Yeah, exactly. But of course, like at some point, like with some species of animals that were living there now, like going back millions of years, like when Africa and South America were still the same, like then those two split off, and then like over time things become very different.
Yeah.
So you separate them at some point things become different, but the timescales we're talking about here is just not enough time.
Yeah.
To make us different enough.
Is there? I think I heard that there's some evidence that modern humans and Neanderthals were on the road to despeciation. Like there are parts of our genome where there seems to be very little Neanderthal DNA, like Neanderthal deserts. Exactly,
yeah. There are some parts in our genome where, even if you look at thousands of humans, there's never any Neanderthal DNA in there.
Yeah.
So what does that mean? Is that something like this is what makes us uniquely human, something like that? Or is it just by chance, or are there some genes that are just it's not genes that makes us human, it's just genes that don't work in a Neanderthal context, but they work in a human context.
Yeah.
Because all genes are like connected, like and work in tandem with each other.
Could there be a scenario where like, first generation, half human, half Neanderthal? Could there be some situation where like the males were able to reproduce but females weren't able to reproduce or something like that? Could there have been?
There might have been, like we don't know.
We can't tell. Okay.
We can't tell. It is possible, but we don't have any evidence to suggest that is in fact what's happening.
Yeah. Would it change our understanding a lot if we did find that first generation Homo sapien and Neanderthal? Would that?
Oh yeah, that would be very nice. Yeah.
Yeah.
Because I think like with ancient DNA, like you can just go back and check. Like we have like if we just look at DNA today, we can make all these models and all these predictions and build our little trees. But having ancient DNA is just so powerful. Yeah. Because it literally just transports you back in time and see, okay, this is what it was.
Yeah, interesting. Did we have? I think I saw a paper, and I think you might have been on the paper too, where it talked about the timing of Homo sapien and Neanderthal interbreeding out of Africa.
Oh, so yeah.
And it was quite a constrained time period, I seem to remember. I mean, there's nuance to this, right? Because this is just one episode of interbreeding, because like you said, the mitochondrial DNA and the Y chromosome show signs
this is talking about the meeting with Neanderthals, that it's something like between 40 and 50,000 years.
Yeah.
Yeah.
What was the time period that you guys seem to do?
We think, so it's also like when we make all these predictions about when we met Neanderthals, we usually assume that there's a single point, and that's the point we report. But of course, like it's probably it's a process that takes time over thousands of years.
Yeah.
So in that paper, we think that the average time is something around 47,000 years, but with a period that extends maybe 6,000 years plus and minus. So an overlap of 10,000 years or something.
Yeah.
With the center on 47,000 years ago.
Okay. So it's sort of a 10,000 year time period that we're meeting, it seems.
Yeah.
And
so like, what is that, 52 to 42 or something like that?
52 to 42. Interesting. How do you square that with like sometimes the archaeological evidence? Let's say the archaeological evidence of when humans arrived in Australia seems to get older and older and seems to predate that point.
Yes.
And they have Neanderthal admixture.
Yes. So, but there's also there's all these things, like it's not necessarily conflicting. Like there could be, like let's say some human stone tools or human skull found in Australia 70,000 years ago.
Yeah.
If that's not the same population, like there's been multiple human migrations out of Africa probably.
Yeah.
If that's not a population that contributes to people today, it will be invisible in genetics because we can only see populations that have contributed to us today. Because we look at DNA today. And we are very biased in genetics. We can only, it's a survivor bias essentially.
Yeah.
We can only see people who contributed.
Yeah.
Because those, by definition, is the only DNA that's around today. It's from ancestors.
Yeah.
Whereas like archaeology can go and find people who didn't contribute.
Yeah. Yeah. And you could only find groups that don't contribute if you have ancient DNA.
Exactly.
Okay. Yeah.
Like Usishim is again like a good example. Like that's a population that didn't really contribute to present day people. But it's because we have ancient DNA, we can say that it is like a human population.
Yeah. This is maybe my last question because I don't even know how I'm going to edit this whole video. Is it possible that groups contributed, but still, just because of recombination and all of these things, we just don't have their DNA within us? Is that a possibility?
Yeah. There is a difference between your ancestors and the genetic ancestors.
Yeah.
Like for instance, if you only looked at my X chromosome, for instance. So I have an X and a Y chromosome. I got the X from my mom.
Yeah.
Like my dad's X chromosome is not passed on to me. Like, but he's my ancestor, right? But like, if you only looked at that part of the region, like he's not my genetic ancestor there. It is one here. So this one actually shows the ancestors, but it's only the genetic ancestors that are highlighted. So for instance, your grandfather is definitely your ancestor, but just doesn't pass on this green piece to you.
Yeah.
Because there are two chromosomes, so there's a 50-50 chance which one you give.
Yeah.
So you have way more ancestors than you have genetic ancestors.
Yeah. I think that's what I'm trying to.
Yeah, that's what I'm trying to explain with the X-ray. I don't think I did it very well. I think it's
yeah, because I just, when we talk about like that population, a population 60,000 years ago that didn't, say it didn't contribute to modern people in a way. Well, it seems like they didn't contribute. Is it possible that they contributed just in a very small way?
And then the DNA just got lost by chance. Yeah. That is completely possible.
Oh, okay. Yeah. Okay. I always wondered that.
That's the bias of genetics. We can only see things that passed on.
Yeah.
That was transmitted.
Yeah. Damn. We're learning a lot. Do you have anything else you wanted to say about ancient DNA, Laurits?
I think it's time for that beer.
I think so. Yeah.
I'm thirsty.
Yeah. That was a lot. That's like an hour and a half of brain-melting genetics talk. Well, before I turn the camera off, I want to thank you for appearing on my channel 3 times now.
Hat trick.
Hat trick. First hat trick. We really appreciate you.
Thank you. Thank you for having me. It's always a pleasure.
No, we appreciate you sharing your time with us because we don't understand, but we want to understand, and we're trying. All right. Cut to us drinking a beer. Cheers, everyone. Cheers. Thanks for watching. Oh, yeah. Cheers.
Cheers.
Hope you liked it. My brain is confused. They liked it. They really loved it.
Yeah.